Chapter 14 deals with vaccines, biotechnology, diagnostics, and therapeutics. So way back in the late 1700s, Edward Jenner was the person who did the first vaccination. He actually noticed that milkmaids didn't get smallpox because most of them had cowpox. So he speculated that somehow the cowpox infections protected them against smallpox. So he actually inoculated a boy with cowpox, and the child recovered quickly, and then he infected him with smallpox, and lo and behold, he showed no symptoms. So it showed that the cowpox actually worked to prevent a smallpox infection. Smallpox really is no longer a disease that we see because it pretty much has been eradicated. And however, it's still recommended to get a smallpox vaccine because you never know. There could be a pocket of this disease somewhere in the world. Now, the CDC estimated that from 1994 to 2013, vaccines prevented 21 million hospitalizations and over 732,000 deaths. I think that speaks for itself, guys. Research is showing that there's a lot of life-saving benefits to vaccines, but a lot of times there's controversy that comes into play with vaccines. It's not surprising that social media has fed into a lot of this. And if you guys take a look back in about 1998, there was a study of just 12 patients, and the paper that was printed in the Lancet claimed that there was a correlation between the MMR vaccine and autism. Well, guess what, guys? After that publications, a lot of people in the US and the United Kingdom decided that they did not want their children vaccinated with MMR. Well, turns out in 2010, this paper was retracted. There is absolutely no link between MMR and autism. And it turns out that the authors were funded by lawyers of patients that had autism, and they were filing lawsuits against the vaccine companies. So there's a huge conflict of interest. So again, guys, please don't believe any of that link between MMR and autism. It's not true. So a lot more studies have been done. And if you take a look, guys, in 2015, 95,000 children that were inoculated showed that there was no link between MMR and autism. Now, sadly, because of this, people are still leery about getting vaccinations overall. And a resurgence of measles showed up in Disney, 2015, 189 cases. And not only did it show up here in Florida, but it did show up throughout the United States, 24 states in particular. And it popped up again last year, guys. Measles is very contagious, and it can be fatal. Now, because there's outbreaks, school enrollment has changed and what vaccines are required, etc. Some states are ruling you can't opt out for any reason, no personal reasons at all. California requires that kids receive at least 10 immunizations to attend school or daycare. The only exceptions are medical exemptions. Now, vaccines can be taken many ways: injected, inhaled, etc. And here's a list of the different types of vaccines that are available out there. So I'll walk you through some of those that should be tested on. So things to consider. Vaccines do not provide you with immediate protection. It takes a couple weeks for antibodies to actually reach a peak. And I want to mention that now, guys, also with the COVID epidemic that has been on everybody's mind. If you get exposed, it could take 2 weeks before any antibodies are actually detected. So you could feel sick, but we may not be able to actually detect the antibodies against this. The good thing with vaccines too, and something that's very important to our immune system class, is that we have to have memory. So when you're vaccinated, you will then have the antibodies. The antibodies in your immune system will recognize the microbe and take care of the infection for you. So recommendations for children, there's more than 15 different recommended types of vaccines. It's used to stimulate optimal immunological memory. And one thing that's very important, if there are boosters required, you have to get those to get the amount of antibody in your system up to where it needs to be. So they're spaced apart because it gives your body time for your adaptive immunity to respond, make those memory B and T cells very, very important against fighting off an infection. We need those. Now, some parents prefer to space out the vaccinations, but this can lead to a breakdown in what's known as herd immunity. Herd immunity is when about 80% roughly has the antibodies. The problem is if the numbers go too far below that, children who are not able to get their vaccinations on time are susceptible. They could get sick, they could actually die. So you have to keep up with the recommended vaccination schedule that's out there. Now, please for your exam know some of the different types of vaccines. Live attenuated, those contain altered pathogens that do not cause disease but are still infectious. Benefits, they stimulate a very potent type of immunological response, and they're accompanied by a long-term memory, so they're actually very good. Again, a big problem with these is that people who are immunocompromised have to be careful. So any of these types of live attenuated vaccines, people need to be aware that they're live attenuated, and in some cases you may opt not to want to have that type of a vaccine. Inactivated vaccines are whole inactivated pathogens. These are better. They can be the whole organism or parts of the organism itself. So a whole agent vaccine, that does definitely have the whole pathogen, inactivated by heat, chemicals, or radiation. Now with subunits, it's pretty cool. They're purified antigens. It's pretty interesting, guys. So they kind of take parts of the infectious agent and they purify the antigens that will stimulate antibodies to be produced. Now, one of the things with these, because they're small, you have to add what's called an adjuvant. And that can be a lipid of some sort, aluminum salts, etc. And that makes it large enough then so that your body will react to it. Now, we're going to go into a little bit of diagnostics now, and there are some essential tools that are needed to identify a variety of pathogens. So a term to know is serology. And serology is the testing of serum. And that's what a lot of the antibody testing now that's going on is involved with. So we test the serum of a patient. Now, quite often we may actually look at what are called agglutination reactions. And in a case like that, one type of agglutination reaction relies on using red blood cells. So please know for your exam and your quizzes, etc., that heme agglutination, heme, blood, is the agglutination of red blood cells. And when we do ABO blood typing, for instance, guys, if you have a person who's type B, they've got these B antigens on the outside, you add an anti-B sera, and what you'll see, this is the agglutination on this side. Really cool. You can see all that clumping or agglutination here. If you were to add an anti-A antibody, though, that doesn't recognize B. Can you see how this is nice and even, guys? So this is how we tell in the blood bank the difference between whether or not the person is B or they're not reacting with anti-A. Another test that's used quite a lot, guys, this was developed a while back, and it's used a lot for different types of diseases. One that comes to mind readily is HIV. So this is the first way that we screen for HIV, but other types of ELISA tests are done as well. So it's an enzyme link immunosorbent assay. And please know there's two types. The first type, guys, is called direct, and that will detect antigen. And the second type is called indirect, that detects antibodies. And you can actually run 96 samples. That's what they're showing here, guys. 96 samples at a time, and you get a color reaction that's read in what's called a plate reader to let you know whether the person has the antibody. And the last test I'd like to talk about is polymerase chain reaction, or PCR. And this is used to amplify up multiple copies of either a gene or just amplify the amount of DNA that's present. I used to work in a forensic lab years ago, and I was there at the beginning of all the DNA typing, and we actually used this to amplify up DNA from small samples of blood, other body fluids, hairs, etc. And it can now be used for actually doing gene sequencing and detecting genetic disorders. So please take a screenshot of this for chapter 14. And one thing on this slide that I will test you on, guys, is that when we are doing PCR, the type of polymerase that is used, please know, is called TAQ, T-A-Q polymerase, and it's heat stable. That's why we use it. Again, TAQ polymerase is used because it's heat stable. And basically what happens, guys, is you start out at the beginning with your strand of DNA. You denature it in the first step by applying high heat. Then you apply primers. And if you remember back when we talked about DNA replication, primers are needed for the DNA polymerase to bind to. Then you copy the strands of DNA, and you continue this for about 30 cycles. 35 cycles, depends on what you're amplifying up. So you get a lot of DNA at the end. So for your exam, quiz, etc., please know these 3 steps for PCR. First step is melting step. You apply a high temperature, 95 to 99 degrees, to separate the 2 strands of DNA. The annealing step, you lower your temperature somewhere between 50 and 65 degrees to allow primers to anneal. And anneal just means bind to the DNA. The last step is extension, and that's usually done at 65 to 75 degrees, and that will allow then DNA polymerase to target, to copy the target DNA.